Hello, my name is Brandon Medina, and in collaboration with the UCLA Sustainable LA Grand Challenge, I'm here to talk about my research project on the effect of cesium chloride on polyp regeneration of isolated Aurelia tentacles. Animal life cycles are generally thought to only proceed unidirectionally, meaning that animals cannot revert back to earlier life stages. Jellyfish, which are part of the Cnidaria phylum, which contains other animals such as corals, hydra, and the Portuguese man-o-war, diverge from bilaterian eggs animals, which includes vertebrates, mollusks, arthropods, and many more, over 550 million years ago. Cnidaria are capable of regressing backwards to earlier life stages. Um, the objective of the study is to develop a reliable protocol which causes polyp tissue to revert back to the stable planula-like state, which is basically a larva state. Um, and then naturally grow back into a polyp state, which will help us define the oral ab-oral body axis. We did this by submerging tissue samples in a 58 millimolar cesium chloride solution for 2 hours, the solution of which helps trigger the settlement of planuloids and thus the metamorphosis back into polyps. This system allows us to study the definition of the primary body axis, which is an ancient development originating with the ancestor of all living things. Definition of the body axis can help us learn more about the evolution of early animals after the origination of neural and sensory systems, but before the development of the mesoderm and secondary body axes. To study the regenerative process of Aurelia tentacles in response to excess cesium ions, we submerge them in this solution for varying amounts of time to study the extent of which these tentacles can regenerate in these conditions. Our design was composed, or our study experimental design was composed of six total trials, one 48 well plate for each trial, yielding a total of 288 tentacle samples total. Five plates were treated with the cesium chloride solution, while one plate was left out untreated, acting as our control. All plates were incubated in the same fridge with simulated day-night cycle and a constant temperature of 16 degrees Celsius for 2, 4, 8, 12, and 24 hours respectively in the cesium chloride solution minus the controlled trial. Samples were allowed to acclimate for 2 days in the incubator before being assigned to a well plate, and every 3 days after the initial day of submersion, we recorded changes in the morphology of the samples with a numbering system over the course of 28 or 20 days from the initial treatment. Currently, we're applying our newfound data into a new experiment utilizing different tissue types, which includes oral disc, calyx, and foot. The procedure remains mostly the same, except all experimental trials are treated uniformly in a 2-hour submersion window. The results of our first experiment demonstrated that the 2-hour submersion window was provided the fastest results with the highest number of metamorphosing individuals by the end of the observation period. In the figure one, we can see that S0 denotes a sample with traits similar to a freshly amputated tissue. Um, S1 denotes an actively moving sample moving via cilia. S2 denotes a sample that has settled and begun metamorphosis. S3 denotes samples exhibiting formation of a mouth pore or a vagination. S4 denotes samples with budding tentacles around the mouth of vagination, and S5 finally denotes a mature and complete polyp with mouth and tentacles that are as long as or longer than the body column, as well as a foot that anchors the organism to wherever it's sitting. Our current experiment is still ongoing, and we do not have conclusive results yet, but we anticipate that out of all of our different tissue types, that the foot tissues will readily return to a polyp-like state the fastest, followed by oral disc fragments with tentacles and tentacle fragments, and finally calyx fragments metamorphosing the last, or the slowest. We can see pictures here. The first one on the very left under results continued depicts a newly amputated tentacle tissue sample with no distinguishable fingers or features. I'm sorry, it's a little bit small, but I can't zoom in. But it's pretty much an amorphous blob. Um, Number two depicts a calyx, which is a body column fragment with a foot-like feature and a mouth of vagination towards the right end of the organism. Number three depicts an oral disc fragment with budding tentacles. It's a little bit hard to see, but you can see there's like a little round bumps near the top side of it. Those are the tentacle buds. And then the last image on the very right, number four, depicts an oral disc fragment again, but with developing tentacles that are much more noticeable and a body column and foot that are less developed. Um, Although our results are incomplete, some progress has been made in determining the healthy level exposure that will help drive the settlement of planula without killing them, because this was a problem for our experiment last quarter. Um, there was heavy tissue death, which was possibly due to overexposure to the cesium chloride solution, as it's lethal if they remain in it for too long. Um, Future iterations of this experiment could experiment with different ways of dissecting the tissues, for example, longitudinally rather than laterally, to see the effect it has, if any, on the time to metamorphosize. Thank you for listening to my presentation, and um, this was brought to you again by the UCLA Sustainable LA Grand Challenge. I'd like to thank them for helping me along the way, and as well as my advisor, Hayden Speck, and my PI, Dr. Jacobs.